So Hodges is here. Hudson is here. Michaela's here in blue. Asbury is here in blue. Midkiff is here. Rice is here in blue? No. Okay. Tudor is here in blue. Massey is here. Burdette is online. Just a reminder, guys, online, you're going to need your cameras on once we get started. It's just my proof that you're paying attention to the lecture and not taking a nap or something like that. Anyway, Van Cross? No. Taylor? Yeah, no Kinsey yet. David? Eric? Gotcha. Eric is physically here. Cedric is online. Richardson? Here. Gotcha. Is that blue or gray? Blue. Okay. Just being sure. But you still get extra credit for being here. Physically being here. Newton? Felder is online. Bowers? Nope. Hemmings, Reed, Taylor, and Van Cross. Did I miss anybody? Everybody's accounted for? Well, it's 8 o'clock. I'll wait until 8:01 before we officially get started. Good job with the people with the cameras on. Thank you. Can you give a wave? I can't see any of you guys. There we go. Nice. Okay, other than Cedric. All right. Let me see here. There he is. All right. So while I'm pulling this up, does anybody have any questions about anything? All right. So again, this is the last chapter 10. Is the last one in the third exam. We're going to finish it soon, so most likely the exam will be next week. And I'm open to suggestions as to when exactly you want to take the exam. So far, I've had one strong vote for Wednesday. So right now, it'll probably be Wednesday unless I get a lot of votes or some strong ones against it. And then we'll figure it out. All right. Almost ready. So start your morning off with a little Jimi Hendrix. All right. Anyway, let me pull up the PowerPoint. So again, exam will probably be sometime next week, probably Wednesday. If you feel different, let me know. Oh yeah. Who has their textbooks on them? If you just raise it up so I can see it all at once here. All right. Okay. Good. Hopefully I'll just have that memorized. All right. So then you guys send me an email saying that you have your textbook, please, and you'll get that extra credit. All right. And if you don't have your textbook and you're not getting great grades, I would recommend getting one. Because like I always say, I teach out of the book because this is a 100 level course. None of the information I give you, or that I test you on anyway, comes from anywhere other than your textbook. It all comes from your textbook. What else was I going to remind you about? Lab this week. It's virtual, so make sure you bring a computer. And again, because it's virtual, I don't care if you come today or Wednesday. You don't need to let me know. It's all the same. What else? So labs, exam, attendance. So I guess the first word for attendance, let's do code again. So even though we did it, I think that was the last word for the last time. The word is code. So again, if you're online watching the video, you're going to need to send me a picture of some kind of code. And the rest of you can just send me the letters or the words. Try to make sure I'm not forgetting anything before I move forward. I feel like I'm forgetting something important. Maybe it'll come to me. Here we go. First thing we'll talk about is the genetic code. We've already kind of talked about it earlier. I'm just reminding you. Actually, let me back it up a lot before we jump into this. Just to remind you the big picture, because I told you my suggestion is to study it this way. First, memorize the fact that we go from DNA to RNA to protein. Memorize that fact. That's what this whole conversation is about. Then once you have that memorized, then memorize the fact that when you go from DNA to RNA, that's called transcription. And when you go from RNA to protein, that's called translation. Then you can start adding in the details, which is what we're doing now. So last conversation we had, we kind of talked about this being a language, right? Like there's the nucleic acid language, and that language happens to have words, just like our language. The English language has words. It also has an alphabet. It so happens that alphabet, right, only has 4 letters. If it's DNA, those 4 letters are R, T, or A, T, C, and G. If it's RNA, those 4 letters are A, U, C, and G. But it's the language with those 4 letters. And those letters come together to make words. And all the words in these languages, in this language, are 3 letters long. And it is, as we mentioned earlier, the genetic code is a triplet, right? We call it a codon, which was on a previous slide. It's not here, but I'll go ahead and try to write it. Yeah, it looks like garbage. But anyway, the codon is what we call it, right? So of the 64 triplets, of the 64 codons, 61 of them actually code for amino acids, and 3 of them say, all right, stop, right? The process is done. And put an X through this, because I'm not going to ask you to know that number. As a matter of fact, I'm not even going to ask you to know that there's 64 words. However, let's think about this. I told you on Friday, well, let's see if anybody remembers, because this is something you need to know. I told you on Friday, and you learned it in chapter 3 when we learned about proteins. How many amino acids are there? Does anybody remember? Yep, 20. It's 20 amino acids. So think about that. And these codes, those words, right? Each one of those 3-letter words says, bring me this amino acid. Bring me that amino acid. That's the purpose of the codes. That's what they're saying. That's what they're coding for, right? So how is it that we have 64, well, 61 in a way, 61 codes that code for amino acids, yet we only have 20 amino acids, right? So just think about that for a while, and I'll give you the answer a little bit later. Not that it's that important for the grand scheme of the semester, but it is something worth thinking about. Anyway, any questions about the slide before we kind of dive a little bit deeper into it? All right. You will get a code, possibly. You might get something like this where I ask you to translate some RNA. And it's actually really simple. Let me show you how to do it. You just start in the middle and you work your way out. So if I say, translate this strand of RNA, and it says, I don't know, GAU. All right, so first here's the G, and here's an A, and here's a U. So that codes for aspartic acid. It's going to be that simple. Does that make sense? So there's nothing to memorize here. In my opinion, you don't even need to practice it. It's a very simple thing. So CCA, right? Here's a C, and here's a C, there's an A, that codes for proline. Make sense? All right, so you'll probably have one question like that on the exam. Most likely, I'll start with a strand of DNA and have you transcribe that into the complementary strand of RNA, and then take that RNA code and use this to translate it and tell me which amino acids it codes for. And it'll be that simple. And of course, also, that's really not that important for 100 level non-biology majors. So if there are any questions, it's probably going to be like 1 or 2. So if for some weird reason this is stressing you out, don't let it stress you out. It's not going to drop your score that much if you mess it up. Also, it's still going to be, it'll still be multiple choice exam. So even if you're completely lost, there is a chance you might get it correct. Also, to remind you what I said on Friday, if I ask you to transcribe something, remember transcription is going from DNA to RNA, right? I'm gonna quit trying to draw on the screen. DNA to RNA is transcription. So if I ask you to transcribe something, and you're looking at the multiple choices, anything that has a T in it is not going to be the correct answer, right? Because there is no T in RNA. You have to have a U. So you can go ahead and eliminate anything with a T in it. So your chances, again, of getting the correct answer are a little bit higher. Anyway, any questions about this? Also, really quickly to point out, I remember I was asking about, all right, how is it that we have 61 codes, but only 20 amino acids? It's because some of these codes, for certain amino acids, there's a few codes, right? So if it's proline, CCU codes for proline, CCC codes for proline, CCA codes for proline, and CCG codes for proline. Of course, we could talk about that all day, but hopefully you see what I'm saying there, right? So for each given amino acid, there's actually a few codes that would work. All right, if there's no questions, let's move forward. This part is where it's really interesting to me. The fact that the genetic code is universal. And I might ask you that on the exam. But yes, it is universal. What do I mean by that? I mean it is shared by every living organism that we know of. So yeah, it's all universal. In other words, too, you would take any DNA from any organism with the proper tools, if you do it properly, and insert it into another organism. Because it's universal, that code could be read, and the proteins will be made. And I'm going to give you the next slide. I'm going to show you some examples of that. But to me, this is where it's really interesting. Like, I just think about all the possibilities, and you can too if you want to for independent work. This is one of the few times. Well, no, I guess you could still use a source, but I was going to say one of the few times you can just think of something off the wall. Remember, like I was saying on Facebook, you could do come up with an idea for an experiment and get points for that. You don't have to perform the experiment, just come up with it. Likewise, I'm telling you right now, get creative. What kind of DNA could you take from one organism to put into another? For example, my brother lives in Seattle. Marijuana is legal in Seattle. I think it would be really cool to take the genes responsible for producing THC and put them in foods that you eat, like tomatoes, for example. So you could have special salsa. I have a lot of other ideas, but I don't want to give them away to you because then you won't be able to have some ideas. So if you can think of some things you want to do, send that for independent work. Now, you do need to look it up just to make sure it's not already an idea. And if it is, you can just say, like, I wanted to see if I can make a salsa that'll get you high. And I looked it up, and yeah, it already exists. Here's the link that says so. Or I want to see if we can get salsa to make you high. I looked it up, and so far nobody's working on that. Whatever the case may be. Anyway, any questions so far about this idea of a genetic code? That's a lot of words on that slide, really, just to tell you it's a genetic code. The genetic code is universal. That's it. And again, there's probably might not be questions about that on the exam. Here's some examples. Can anybody tell what's different about these pigs? Because it's kind of hard with the lighting and all that. Can anybody tell what's different with those pigs? Or that one in the middle, I guess. Yeah, specifically, and it's hard to tell in that picture, it glows. That pig glows. So they've taken the proteins from jellyfish that make them glow, and they inserted them into that pig, and now the pig glows in the dark. Same with that rat or mouse. I forgot which one it is. A little baby rat, baby mouse, and it literally glows. And we actually do this a lot in science, not just for the sake of having things glow, but a lot of times if you want to see how proteins are expressed, we use jellyfish proteins because obviously if whatever protein we're looking at in an experiment starts glowing, we'll see it, right? So that's all we do that. This is not a real example, but I read about it and people talking about doing it and wanting to do it, putting the jellyfish genes for glowing into plants so that the trees glow. So you don't have to use lights, street lights. So again, that's just a hypothetical. That one's not real yet, but these other are. And if you want to, you can look that up for independent work. What are some other things that glow now, thanks to scientists? And then there are some other examples too you can look up. for independent work. Just again, this idea of, well, basically GMOs. So think about, I think it's called golden rice or super rice, something like that. Has anybody ever heard of that? Everyone's afraid of GMOs, but they're really not that scary. So, for example, this golden rice or super rice, I forgot which, they took the genes that are in carrots that make them give you vitamin A, the good stuff. It's good for you. They took those genes out and they put it in rice. So this rice now produces vitamin A. That's it, right? And there's things like there's a certain kind of apple. I forget what the name is. Anyway, the genes that's responsible for making them turn brown when they get exposed to air, they just cut those genes out. So it's not always that scary. I get some of it, like some of the stuff basically has its own built-in pesticide. I can see why that scares people, but I don't see what's scary about like pulling a gene out, for example. Anyway, any questions about this? Really fascinating stuff if you're into it. Look into it for independent work, but for the grand scheme of the semester, it's not all that. Anyway, now that we have all the background information, now we can finally start really jumping into it. So let's talk about the first process, transcription. As you already know, and the short way to write what's on the top bullet point there is transcription is DNA to RNA. That's what you need to know. You need to know transcription is going from DNA to RNA. And to help you remember that, again, like I said earlier today and on Friday, when we're going from DNA to RNA, it's the same language, right? It's that nucleic acid language where it's 4 letters in the alphabet and 3-letter words. It's all the same, right? Just like any other situation, if you were to write something down in the same language, you're just transcribing it, right? Actually, that's what you guys are doing right now. I see your hands moving. You are transcribing something. It's the same language. We're going from English language on the board to English language on your paper. You're transcribing it. As you already know from the previous slides on Monday or Friday, the DNA strands separate where the process starts. So we already talked about that, right? Because that's where the code has to be read. So you need to open up the DNA strand. Not that I'm going to ask you that, but again, I teach out of the book. Once that DNA strand is open and the codes are visible, I guess, or accessible, one of those serves as a template for the RNA. The other one's just completely not used. And then the enzyme, which we're going to talk about here in a second, it adds each nucleotide one at a time. So this is everything I've already told you on Friday, except now there it is in writing for you. The next word for attendance is going to be in, like this. So if you're online watching the video, the first word, remember you needed to send me a picture of that. This one, I need you to take a screenshot of me showing you pointing at the pin. Remember your entire screen, though. Don't zoom in on it. I need the entire screen so I can make sure it's your screenshot. Anyway, any questions? All right. As we already know, again, this is still a review. This is the next bullet point. The RNA nucleotides follow the same base pairing rules that we've already talked about. So I'm going to try to write this on the board. Again, it's probably going to look like garbage. C always goes with G. A's always go with T's. But in the situation where we're talking about RNA, there is no T. There's a U instead. So nothing new there, right? Here's some new information, sort of. This is the first time I'm officially telling you about this, but I unofficially told you on Friday. The enzyme that does this is called RNA polymerase. So there you go. Now you've been introduced to all 2 enzymes that will be on the exam. DNA polymerase makes DNA, like we said last week. RNA polymerase makes RNA. That simple. Or to help you remember it properly, I guess we could say DNA polymerase is the enzyme that makes the DNA polymer, right? We don't even say it makes the DNA. We get even more specific. And RNA polymerase is the enzyme that makes the RNA polymer. So hopefully that'll help you remember the full name. And this is what it looks like, sort of. So here's a strand of DNA. The enzyme is clamped on, it's reading the code, and it's making a strand of RNA. And we're going to talk about that process in more detail. Also, it's worth noting, just like the same thing I said for DNA polymerase, your book calls this RNA polymerase. Well, that is what it's called, but your book kind of teaches it like it's just one enzyme. But in all actuality, it's a team of proteins coming together. So if you want to look that up for independent work, you can to make it more complicated. Otherwise, we can just study this easier to remember version. So any questions about RNA polymerase? Okay, there's another picture of it. Same thing. This is everything I've just said, except now it's in a drawing, so you can see it happening. Like I said, the DNA opens up, so the code is read. One of the strands serves as the template. The other strand is not used. In this particular picture, this strand is serving as a template. It's just reading the code and bringing in each of the individual nucleotides. So this one says T, right? So it brings in an A, and that's going to get covalently bonded to this. And this one's an A, so this one's going to bring in a U, and it's going to get covalently bonded to that one. And again, another A, so we bring in another U, and it'll get covalently bonded. And you can see you're just making the new mRNA strand longer and longer, one at a time. It really is, well, we really do teach it that simply, right? As far as you're concerned, it is that simple. And again, it's the RNA polymerase that does this. Any questions about this? I'll remind you for the Friday, I think I had an idea for independent work. Whether we're talking about DNA polymerase or RNA polymerase, the question I posed to you was, where are these nucleotides coming from? They're just floating around in your cell, right in the cytoplasm. Where do they come from? Do your cells make those nucleotides? Do you get those nucleotides by digesting DNA that you've eaten? You can look that up if you want for independent work. Any questions about this slide? All right. So here's where it gets a little bit more complicated. Remember, this whole process of going from DNA to protein, that whole process, right? The first step of that whole process is called transcription. That's going from DNA to RNA. That's what we're talking about later, or right now. Later, we're going to talk about going from RNA to protein. So this one step of transcription itself is broken down into 3 stages, and you need to know them, and you need to know them in this order. Initiation of transcription, RNA elongation, and termination of transcription. We're going to talk about each 3 separately. I bolded the parts that you can remember. So if you like to take notes or make note cards to study, it'll be sufficient to memorize the words initiation, elongation, and termination. For the multiple choice exam, I'm not going to try to trick you and say something like initiation of transcription or initiation of polymerization, right? I'm not going to try to trick you. So it's sufficient to memorize initiation, elongation, and termination. And when you think about it, that's pretty simple, right? Logically, those words make sense. You start something, that's called initiation. The whole point is to get a strand of RNA, so obviously elongation, that makes sense. And then termination, obviously, is the end of something. So it is actually quite simple, at least as far as memorizing the correct order. So any questions about that? Okay, let's talk about it. Let's talk about each one of them individually. Now I have some more words that you need to know for the exam. The promoter, that's where initiation starts. And remember, initiation is the first part of transcription, right? So initiation starts at something called the promoter. That one's a little bit, not hard, but it's not as easy as initiation, elongation, termination, right? Those words, this has to be that, right? Here, promoter, that could be a lot of things. But I'm telling you now, that is where it starts. Promoter is where it starts. You need to know that for the exam. So what is initiation? Simply put, it's when the RNA polymerase attaches to the DNA at the promoter. That's it. Pretty simple, right? The machine that does the work, it attaches at the right spot, which is what we call a promoter. That's it. That's what initiation is. I still see people taking notes, so I don't want to move too fast. So some things you can look up if you want. What exactly is a promoter? Because obviously we're talking about a DNA strand. So what makes a promoter a promoter? If you don't need to know it, If you can, look it up for independent work if you want. Is it a certain code that the RNA polymerase recognizes? Like, oh, yeah, that's a bunch of A's. That's what I'm looking for. That's what I'm going to attach. Is it the same for every gene, or is it different from some genes? Are there ways to turn it on and off? It's important too. And we'll kind of hint at that one a little bit later. Anyway, any questions about this slide? It's a lot of information on that, in those words, just to say initiation is when RNA polymerase attaches to the DNA in a place called the promoter. That easy. Okay, moving forward. Like I said, the next step is elongation, or excuse me, elongation of RNA. But again, like I said earlier, you can just memorize it as elongation. And that is exactly what it sounds like. The RNA polymerase is moving down the gene, it's moving down the strand of DNA, it's reading the code, and it's making a strand of RNA. It gets longer and longer. Oh yeah, what I haven't mentioned earlier, not that this is important at this level, but yeah, and you saw the pictures already. The DNA does come back together. So yes, it opens up so we can read the code, but then after the code is read, the DNA comes back together. Not that that's important, but yeah, there you go. So any questions about elongation? Quite simple. Man, it's quiet in here. You feel like it's a Monday, 8 in the morning or something. All right, the last part, like we already said, termination. And this one's really easy to remember. Where does termination happen? At the terminator. Not actually like the robot from the movie Terminator, but I had to throw it in there. It's even more relevant now with AI becoming the way it is. Anyway, yes, so the third phase is of transcription. The last phase is called termination. Termination is when RNA polymerase hits the terminator sequence, and then it knows that its job is done, and everything disassembles, right? The RNA polymerase goes one way. The new RNA that we just made, it goes another way. The DNA is put back together, and all three parts they're all separated. And it really is that simple. This picture's not from your textbook, so again, you don't need to worry about 5' and 3'. For you guys, that doesn't matter. And again, I'll ask you if you want to look it up for independent work, this idea about a terminator. What is it about the code and the DNA that says, all right, this is where RNA polymerase needs to detach? So what is it, a certain code? Is the code universal? Like, are all terminators the same code? Or are they different for each gene? Are they different for different organisms? All stuff you can look up if you want. There's a question on your study guide that might be on your exam. Should be pretty simple. Let's see if anyone can guess it. What would happen if there was a mutation to where you no longer had a terminator, right? The code, whatever code it is there, it got mutated, and then no longer, the terminator is no longer a terminator. Well, of course, it's easier if it's multiple choice. So I guess if no one wants to take a stab at it, I'll tell you. Basically, the RNA polymerase would keep going, and we would just have a really long strand of RNA. So yeah, that's definitely a question on your study guide, if I remember correctly. Maybe a study, or maybe a question on the exam. All right, any questions? Oh, that's what I was going to remind. That reminded me, speaking of exams, that was the other thing I wanted to remind you of. With the exam coming next week, I want to remind you that once we take exam 2 or exam 3, it is then too late to retake exam 2. So if you haven't retaken exam 2, you should. Even if you just go in there and blindly just take a stab at it, I mean, that's almost a waste of time because most likely your grade's not going to go up from that. But remember, your grade can never go down. Even if you somehow get a worse grade, we'll just keep your original higher grade. Anyway, any questions about the exam thing? And also as a reminder, since you can no longer retake exam 1, you can now come to me and I will sit down with you and talk about every single question you missed and tell you why it's wrong, and maybe even some of your correct ones. So I can talk about why that one's correct. And if you do that, you get a 5% boost to your grade for that exam. And obviously, you're going to need all those correct answers because I use your old exams to write your final exam. So your first 3 exams are your study guide for the final. And if I were you, I'd already be studying. Anyway, this is it. This is everything we talked about, one gigantic picture. There's the DNA strand up there, right? You can see the promoter on the left, the terminator on the right. This is initiation when the RNA polymerase attaches itself to the DNA at the promoter. Elongation, it's just moving down the code or moving down the DNA, reading the code, making the strand of RNA. Then finally, the RNA polymerase hits the terminator, and that tells everything the process is over. So all three parts go their own way. Very simple stuff, right? At least the way it's presented here. Initiation, elongation, termination. You need to know it in that order. And again, in my opinion, it should be easy to remember what happens in each of those because it's in the name. Also, logically, it just has to happen that way, right? The machine has to attach, has to read the code, has to detach. But if you download the PowerPoint, you can watch this little animation that shows everything I just showed you in action. All right. Now, we're sort of done with transcription because remember, transcription is when we go from DNA to RNA, right? And I just showed you how we go from DNA to RNA. But it gets a little bit trickier, so we can't go straight to translation. First, we have to have a little bit of in-between information. And we're going to talk about the processing of eukaryotic RNA. So here we go. I'm not going to ask you any questions about prokaryotes. The point here is that your book is just making the point that if you're talking about bacteria, for example, versus humans, for example, the process is a little bit different because prokaryotes don't have any organelles, including nuclei. So their DNA is just out there in the cell, right? Therefore, it makes sense for prokaryotes to read the code, and then that RNA is then put to use right away, right? That RNA is being translated right away in bacteria. We are a little bit more complicated. Our DNA is inside of a nucleus. So that RNA needs to be processed before it can leave the nucleus. And once it is processed, it is called mRNA, and you definitely need to know what mRNA is. I'm going to be teaching you about, let's see, mRNA, tRNA, rRNA. In this chapter, you're going to be introduced to 3 different types of RNA, and you're going to need to know which is which for the exam. And then later chapters that we're actually going to skip, they teach you about other types of RNA. You can look into that for independent work if you want. Anyway, so again, you need to know what mRNA is. That is the messenger RNA, which makes sense, right? Because we're transcribing the message. We have this code written in the DNA. Well, we transcribed it into the RNA. The message is now in the RNA. So any questions about what mRNA is? All right. You guys are pretty easy today. So, like I said, the RNA can't just leave the nucleus. It's got to be modified. And what we're going to do is talk about these modifications. The first modification is something called cap and tail. And you need to know this. And basically all it is, it's sort of like it sounds. We add a cap to it, we add a tail to it. This picture's not from your book, hence the 5' and 3' stuff. Again, you don't need to know that. But if this was a Harvard class, you would need to know that the cap is on the 5' end and the tail is on the 3' end. Another thing, if I remember correctly, your book doesn't even call it the poly A tail, which is what it is. I'm just requiring you to know it's called the tail. But yes, technically it's a bunch of A's. And you might be wondering why. Why do we do this? Well, there's 2 main reasons. First of all, as you're really going to learn later when we talk about viruses, you don't want a bunch of genetic information just floating around in your cells, right? Not good. It's how viruses get spread. So a lot of times, genetic information that's just floating around in your cell gets attacked. There's a good independent work topic for you too. What does that? What detects foreign genetic information in your cells? How does it destroy it? How does it know it's foreign? Anyway, so that's one of the reasons we do the cap and tail, to prevent that from happening, for protecting it. The other reason is, I'm going to teach you later, there's something called ribosomes. And yeah, it's right up there now, but I'm going to teach you more about it later. Ribosomes are the thing that do the work, so to speak. They're the things that are actually going to do the translation, right? So we know RNA polymerase is the thing that does transcription. And I'm going to teach you later, ribosomes are the things that do the translating. So in order for that machine to recognize that DNA, it needs the cap and tail. So any questions? I guess cap and tail can be the next word for attendance. So if you're online watching the video, send a picture of a cap or a tail. If somehow you have a picture of both, go for it, but just really one or the other will work. For those of you who are in person and maybe sending me those words for extra credit, you can just send me the words, obviously, because you're here. I don't need the picture. Anyway, any questions about this slide? All right, that's one type of RNA processing. Let's talk about the other one. The other one's called RNA splicing. And basically, this is where we get rid of, and I'm going to use quotation marks here, junk DNA. I'm using quotation marks because it's not quite junk. It does serve a purpose that we don't talk about in this class. Another good independent work topic if you want. So yeah, that's why I use quotation marks. It's not actual junk. As far as we're concerned in this class, it is kind of junk. And here's the hard part about RNA splicing, in my opinion, is knowing which part is which. Introns versus exons. So I'm going to teach you the right way. Exons are the parts that get expressed. That part of your genes get expressed. Those are the parts that basically are going to code and be read when we make the proteins. Introns are the parts that are in between. If you remember it like that, you should be okay. What always made this hard for me as a student is I think of exons, I just think of exit. So I think that's the part that we're getting rid of. But it's the opposite of that, right? Exons are the part we keep because that's the coding part. That's the part that's expressed. Introns are the parts that are in between those. So you can see here in this picture, which is, again, is not from your textbook, we have all the introns in blue, the exons in red. So we remove the introns before it gets translated. And I'm not even telling you why. You can look that up for independent work if you want. Why do this? So you know why there's a cap and tail, right, for protection. But why do this? Why have the introns in the exons? Why remove the introns? But yeah, for our class, as far as we're concerned, it is just that simple. You just need to know this is one of the things that happens to RNA. You don't even need to know why. You need to know in this situation, exons are kept, and introns are gone. Any questions? Okay. Oops. Yeah. So your book does point this out, and I like this. This to me is very interesting. RNA splicing plays a significant role in humans because it allows our about 21,000 genes to produce many more proteins. And again, if you're a thinker and you're really thinking about this and not just letting it hit your eyeballs or in one ear and out the other, right? If you're really thinking about this, hopefully that would you wonder, like, how is that possible? How do we have only 21,000 genes, but thousands more proteins when genes are the instructions on how to build proteins? Like, how is that possible? And that's what we're about to talk about on the next slide. But another good independent work topic too. Exactly how many genes do we have? Of course, that number changes as technology changes and we have more information. But at the time that this book was written, there were about 21,000 known genes. So you could look it up and see what it is now. Also, how many proteins or polypeptides do we produce? That also is a number that changes as medical or as technology gets better. So I'm about to answer this question. How do we only have 21,000 genes, but many more polypeptides? I'm about to answer that question. But before I go to that slide, does anybody have questions about this slide? All right. Well, it's not the slide yet. I'll answer that question on the next slide. This is just a picture from your textbook that shows you this whole process of RNA splicing, right? So in this picture, the exons are still red, but the introns are like a lighter color. So you can see we just sliced it out and put it together. Oh, another independent work topic, because your book doesn't teach it, so neither do I. But what is doing this? What is the machine, so to speak? What is the enzyme that's responsible for cutting out these things and then bringing it back together? Because that is literally what's happening, right? The RNA is getting cut and then spliced back together. Anyway, let's go back, or let's get to answering that question. Something called alternative splicing is how we have 21,000 genes, but many more polypeptides. So here we have this one gene. This picture's not from your textbook. It's been simplified, so there's what, 5 exons and then a bunch of introns. Again, another independent work topic. How many exons is the average gene, right? Because here, this one only shows 5. I guarantee you there's a lot more than that because this is just simplifying. So yeah, you can look that up for the moment. How many exons does your typical gene have? Anyway, back to this example. There's 5 exons here. The way I taught it earlier was we get rid of all the introns. And that is sort of true. That's what's happened here, right? We got rid of all the introns. That's gone, that's gone, that's gone, that's gone. All that stuff was spliced together. So now we have exon 1 attached to exon 2, attached to exon 3, attached to exon 4, attached to exon 5, right? That's the way I just taught it earlier. But I'm telling you now that there's alternative ways of doing it. So in this case, not only did we get rid of all the introns, but we also got rid of exon number 3. So all the other ones came together, but exon 3 is not there. And in this case, we got rid of all the introns, but we also got rid of exon number 4. And you can see these different codes then produce different proteins. And of course, those pictures probably don't make much sense to you, but that's fine. If you remember from chapter 3, a protein's function is based on its shape. And you can see just by looking, even though those probably don't make any sense to you, they have different shapes. Therefore, they have different functions. All that by doing some alternative splicing. So again, here we have this simplified version of alternative splicing, and we have one gene that is now coded for 3 different proteins. Anyway, yes, a lot of alternative or independent work topics too. Other thing you could ask, because I asked you earlier, I said how many genes is it, or how many exons does a typical gene have? We could also look this up. How many bases? How long is the typical exon? Because you can see here, exon 1 is pretty long, exon 2 is pretty short, exon 4 is really short, exon 3 is a little bit larger than exon 1. So as you can see, they are different sizes. So you can look that up. What's the typical size of an exon? What's the largest exon that we know about? What's the smallest exon that we know about? All those things. Anyway, any questions? All right. Nice and easy. That's it for transcription. Remember, transcription is going from DNA to RNA. You now know that. Now let's talk about translation. And this will be good. Your book breaks it down into two discussions, basically. So before we talk about the process of translation, first your book introduces you to the players, so to speak, the parts, the machinery, if you will. Probably perfect. It'll take probably about 10 minutes to introduce you to that. And then when we get back on Wednesday, we can talk about how they're all put together. So here we go. The first important ingredient is the mRNA. And you've already been introduced to that, right? You know that. mRNA, that's the messenger, right? That's the message. That's the thing that says this is how you build the protein. This is the thing, that's the code, right? So obviously you're going to need that. Next thing you're going to need, and we're going to talk about it later, because I'm going to give you the name for it. We're going to need the machine that translates it, right? Because that's the process we're talking about is translation. So we need to talk about the thing that does the translating, and we will a little bit later. Going to require some energy and some enzymes. I'm going to put an X through that because I just told you about it, but that's the last we're going to talk about it. Just, yes, we're going to need some enzymes. We're going to need some energy, but we're not going to talk about it. Just know in the back of your mind that it's needed. Not going to ask you about it. So the other components, I think I just need to fix this slide. That's the problem. Because the ribosome, that is the machine that does the translating. So the same way DNA polymerase is the thing that makes DNA, and RNA polymerase is the thing that makes RNA, ribosomes are the thing that translate. Ribosomes are the thing that make the protein. And yet another type of RNA that I told you there'd be 3. So here's the second one, transfer RNA. And I'm going to teach you more about transfer RNA. For now, I'll just tell you, transfer RNA transfers amino acids. And again, I'll teach you that later. Now you know for now. So those are the parts that we're talking about for now. mRNA, ribosomes, and tRNA. Those are the important parts as far as the exam is concerned. So any questions about that side? All right. So again, this whole process is going to require an interpreter because it's translation, right? When you translate something, you need an interpreter. And that, again, is the ribosome that we're talking about. It converts the three-letter nucleic acid words, as you already know, those things are called codons, into amino acid words. So again, that's why I mean it's a translation. It's not transcription, because we're going from one language to the other. We're going from nucleotide, nucleic acid language, to the acid of amino acids or proteins, however you want to say it. And like I said earlier, there's this new type of RNA. It's called tRNA. It's the thing that's transferring the amino acids, right? Because that's how we're building, right? We're building a protein. So we just need to attach a bunch of amino acids to each other, and transfer RNA is the thing that's bringing them over. And yes, technically, and we'll talk about this later too, technically they match the amino acids to their codons, right? So there's a different tRNA for every amino acid. They're very specific. And we're going to talk about that again a little bit later. I'll show you a picture. You can learn how it works. But for now, just know that tRNA is the thing that brings in the amino acids. Because again, that's what we're doing. That's what translation is building a protein. We're just stringing a bunch of amino acids together. If something needs to bring them in, it's the tRNA that does that. And again, like I just said, and you can see it right here in writing, basically this is just saying the tRNA are very specific. And they all, each tRNA, or let me say it the other way around, each amino acid has its own tRNA. It recognizes, that type of tRNA recognizes that type of amino acid. And obviously, as we keep hinting at throughout the semester with this whole structure function thing, it's the unique structure that enables them to do both. I'll put it next to that. That's just trivial. And yes, we will actually talk about that, but I can ask you that. With the exam, again, you need to know that tRNA is the thing that's transferring the amino acids to this whole process. We're going to talk more about this, but are there any questions so far? All right. Here's a picture of it. This picture is actually not from your book. This is just for your own knowledge. I'm not going to ask you any of this. Again, what you need to know for the exam is that tRNA is the thing bringing in amino acids. But let's really quickly talk about how it does that, how it's made. It's a single strand of RNA, which it might not look like in this picture, right? That looks like a double strand. That looks like a double strand. That looks like a double strand. So does that. But the reason it looks like that is because that one strand of RNA is kind of folded in on itself. But it has formed some regions, like this right here, the anticodon loop, where it recognizes the code on the mRNA. And on this other end, this is where it attaches to the amino acid. And this will make much more sense when I show you the actual picture from your textbook. So the picture you're seeing now is a more realistic version, but it also has a lot of information you don't need for this 100 level course. And the picture I'm going to show you, I think on the next slide, is a very simplified version of this. Is everybody ready for me to switch and show you the next one? Again, as far as the exam is concerned, this is almost a useless slide. What you need to know for the exam is tRNA, or it's the thing that's transferring the amino acids. Yeah, I already showed you that. On one end is the anticodon. That's the thing that reads the code. On the other end, that's where the amino acid attaches. And there's a picture of it. Right. So there's another drawing of, but this one's from your book. You can see it's a big squiggly mess. But this is better. As far as you guys are concerned, this is better, right? The tRNA attaches to the amino acid on one end. On the other end, it has a code or an anticode where it reads the message. That's how it recognizes it. This is how it recognizes that it needs to come and attach to the mRNA. And that's it. And again, for the exam, the only thing you need to know is that tRNA is the thing that's transferring the amino acids. Any questions about this side? All right. Now let's talk about the ribosomes. Like I already told you earlier, ribosomes are the translators. That's the thing that's actually making the protein. This is the thing that basically brings all the other stuff together. Yes, technically it's an organelle. I'm not going to ask you about that. It coordinates the functioning of mRNA and tRNA. Or, as I said just a few seconds ago, it's the thing that brings all this stuff together, right? It is the thing that's attaching one amino acid to the next, which is how we build a polypeptide. Like I said, when we first started this chapter, as far as I'm concerned, when we're in this chapter, polypeptide and protein, they mean the same thing in this chapter, right? Because remember, a polypeptide is a strand of amino acids, and then once it gets folded into its correct shape, then we call it a protein. As far as we're concerned right now, it's the same stuff. Here's where it gets a little bit trickier. I'm not sure if I'm going to ask you this or not. Probably not. The ribosomes are made up of 2 subunits. So basically, it's made up of 2 parts. That will be important when I explain how it works. But again, I don't think I'm going to ask you that on the exam. So I put a question mark right there. What you do need to know, like I already said, is this 3rd type of RNA, which is ribosomal RNA. So there, now you've been introduced to all 3 types. Messenger RNA, which is the message, right? That's what we make when we read the code on the DNA, right? When we do transcription, that's what the RNA polymerase is making, is the mRNA. Then I introduce you to the tRNA, which is the thing transferring all the amino acids in. And now finally the 3rd type, which is called ribosomal RNA, and that's the stuff that makes up the ribosomes. And I'll put another question mark next to this. To fully understand this process, really the most important thing you need to know is what mRNA and tRNA, what they are. To me, this is trivial at this level, at the 100 level. The fact that ribosomes are made up of rRNA is kind of trivial. Plus, then there's, again, the process of elimination. I said you definitely need to know mRNA and tRNA. If you know those 2, then my process of elimination, the other one, is the one that makes up ribosomes. Speaking of the 2 subunits, and I'm definitely not going to ask you this. Of course, we will come back to it. This is going to be part of the story of explaining how it works. They have a binding site for the mRNA on the small subunit. So again, the mRNA is going to attach to the smaller part, and the tRNA attaches to the larger part. Not that I'm going to ask you that. Any questions about this slide? That's what they look like. So you can see the two subunits, the small part, the large part. Also, I'll tell you this. This is another simplified version. If you look at another book, a ribosome might look a little bit different, because there's more than 2 sites. In all actuality, there's more than 2 sites, but your book is trying to simplify it. So maybe the last word for attendance will be blue. So you guys can just send the word blue. But for those of you online, send a picture of something blue. And again, I'm going to call an audible like I did last time. If you're online, make sure you get those pictures to me by, let's say, 8:55. So you've got 5 minutes. Any questions? All right. I'll see you in lab, or maybe not. It's up to you. Have a good week.